Hello, everyone, and welcome to today's webinar, Development Advice for Gene Therapy Products, presented by Roe. I'm Joe Keenan, contributing editor to Fierce Markets, and I'll be moderating this webinar. Our speakers today are David Shoemaker, PhD and Senior Vice President, R&D at Roe, Dana Minnick, PhD and Senior Research Scientist at Roe, and Scott Burrian, PhD, Senior Research Scientist at Roe. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you'd like to download the slide deck, please click the Resources and List button at the bottom of your screen. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. You can find additional answers to some common technical issues located in the Help button at the bottom of your screen. The webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentation with a Q&A session. Please submit your questions using the Q&A button on the left side of your screen, and you can do that throughout the presentation. All right, I think that covers it. We're ready to begin. David, please go ahead.
Thanks, Joe. I'm afraid I'm going to have to start off with an apology as Jamie Chang, our medical officer, is serving jury duty and was unable to join us. So I'm going to cover his portion of the clinical aspects of this talk. But I'll get started with a background on the current status of gene transfer. And we prefer to use gene transfer as opposed to gene therapy because these are generally investigational products and they're not therapies as yet. Then I'll cover clinical development strategies, turn it over to Dana for the non-clinical development strategies, and then finally to Scott for the CMC and quality development strategies. And then finally, Dana will circle back with some regulatory programs that are available for developers of these products. So as far as the current status goes, to date, there have been over 2,500 gene transfer studies conducted, but only a few products have been approved. So what that says is that most of these clinical studies were not successful. Hopefully, the people that conducted those studies learned something in the conduct of those studies. But unfortunately, there's been a lot of work done. I've been doing this since the late 90s, and I've been frustrated with products several times. So we're looking to change that trend, hopefully. We feel that gene transfer is at a tipping point potentially, but it's going to take a lot of hard work. It's going to take a lot of collaboration. I put in an URL here for accessing a journal of gene medicine, and I want to emphasize that you can't work in this field without being very up to date with the science. The science is changing on a month-to-month basis, and if you don't have the latest techniques, you're going to suffer and fall behind. So it's a close collaboration between academia and the pharmaceutical industry or biotech. Pharma has lost a lot of its scientists, and consequently, a lot of these technologies are being developed in academia where the academics don't understand drug development. So it's got to be a coordinated team accessing both science and drug development for your program to be successful. This just shows you the number of studies that have been done over the years. Obviously, a lot has been done without much result. They also are focused primarily on early phase studies, phase 1 and phase 1/2. Gene transfer is such an exciting field that these products are going to move quickly to approval if they work. The problem is there's a lot of loose ends to tie together before you can convince FDA to approve your product. I think the phase 1 and phase 1/2 studies and phase 2 have a large row to hoe with clinical pharmacology, dealing with issues like persistence of the transfection and also just how the immunogenicity of these different vectors affects the distribution of the product, and how durable is the gene once it gets transferred. So there's a lot of questions that need to be answered early on before FDA is going to even talk to you about efficacy. I think cancer has obviously demonstrated the most success in moving potential therapies forward, but there are other areas obviously being studied, and they will continue to be. The easiest thing to study is a disease where there's one protein missing or one gene defect. That allows you to focus in very quickly on your therapy. If you have a rare disease where you don't understand the mechanism or the number of genes involved, you have an extremely difficult task getting to the therapeutic stage. A number of vectors are used. I feel like we're at a tipping point now with gene transfer development, but I felt that way before. I felt that the advent of plasmid DNA was going to simplify gene transfer development, and that didn't prove to be the case. So please understand that there are a lot of variables, and if you don't have all of them well controlled, you're not going to be successful, as indicated by the number of trials. Recently, FDA has updated its guidance for the development of these products, and I think everyone's aware that long-term follow-up is necessary. Manufacturing is an issue. Distribution of manufactured products, especially the cell-based products, is a very tall task and an expensive task. And I think one of the advents of gene transfer guidance that I find encouraging is that they're getting down to specific diseases. I think that in the past has been a problem. It's been problematic in other development of products like biosimilars. If FDA provides general guidance, it's really not that helpful. They really have to give developers specific guidance. FDA no longer has laboratories. So all the work is being done in the industry's laboratories, and the FDA is really serving the purpose of being a synthesizer of information, and they can provide value in their crafting of these guidances to the developers, whereas they can't really tell you what science to do if you don't understand it yourself. So with that, I'm going to move on to clinical development strategies and just obviously acknowledge that there are unique aspects to gene transfer clinical trials as opposed to more traditional drugs and biologics. Obviously, you need to be aware of all the guidance that FDA is providing, and I'm going to cover some of the challenges to being successful in getting your product to market, which few people have been able to do. The whole concept of phase 1, phase 2, phase 3, I think is going to go out the window with these products, as it has started already to do with rare diseases. You're going to be in negotiations with FDA more frequently with these products, and if you don't go to FDA with your plan very solidly laid out, FDA is going to push you in directions that answer some of the questions they're asking rather than a straight line development to approval. As I said, early phase studies are the rule rather than the exception. There are questions that need to be answered around feasibility, but primarily dose exploration. Early efficacy is a desire for all people developing these therapies. Many times they're in life-threatening diseases, but if you don't focus on dose exploration, even a preliminary efficacy result is not going to be of much value. So as far as the different vehicles that are being used, there's myriad to choose from. In addition, there are some that are delivered systemically versus locally. Obviously, it's a little bit easier if you have a local application, say in a tumor or in a ocular environment, and that simplifies it. However, there are cell therapies that are being administered systemically, so that's a viable path. The starting dose is determined primarily from your animal studies, but it's still an experiment in humans from my perspective. You're going to have to monitor the first subjects that you administer your product to very closely and be very cognizant of the potential for immunogenicity. And at this stage, the animal studies in many cases are not any more valuable than in vitro studies. Your understanding of the mechanism of your disease that you're treating and the extent of the integration of your gene is probably the most influential thing determining the dose. Early efficacy data, this is probably more problematic than in other types of product development programs simply because you cannot use surrogate endpoints to any degree. They're really scientific surrogate endpoints that you're using. FDA, as you know, is all about clinical benefit. And the fact that you're able to get integration or you're able to get copies of a particular protein is a good scientific result, but it's not efficacy to any significant degree. It's more pharmacodynamics. You can also, for the specific diseases like retinal disorders that have assays that allow you to assess efficacy soon, that's excellent. And those things like fluorescein angiography can be used ultimately to support your filing of your marketing application. The later phase trials, like I said, these may actually go away to a large extent in the environment of adaptive clinical trials. I think you're going to be dealing with fewer patients in your database. The rare diseases with life-threatening gene mutations are going to have extremely low numbers of subjects. So once you get your program started, you should be able to amend your protocol and get the data sufficient to get your product approved with hundreds or even less than 100 subjects. And that's been the case in the products that have been approved to date. Adverse event monitoring, this gets back to the importance of having an understanding of your disease and the mechanism of integration of your gene. Long-term follow-up will be a very important commitment that you make. 15 years has been suggested in some cases, but I think putting subjects in lifetime databases for following is not unreasonable in this day of using electronic health records and monitoring post-marketing approvals. That's going to become more the rule rather than the exception with these extremely novel therapies. And gene editing is on the horizon as that's just coming into its own in the non-clinical arena and should be being administered to humans in the near future. As far as durability of effect, I think we're familiar with the different products that have been able to replace very small portions of the missing protein, such as in hemophilia, and yet get some efficacy, which is important. But how long that efficacy lasts can be determined by such things as the immunogenicity of the vector and other concerns about the viability and the durability of the cell that you've transfected. So FDA's guidance in the disease areas are specific to hemophilia and retinal diseases at this point in time. One would hope that FDA would apply this focused approach to guidance for other diseases that are able to be treated with genes. Hemophilia guidance is relatively generic. They have, in my opinion, unfortunately come down with the suggestion of a non-inferiority trial, which, as most of you who have been in this business a long time know, is not the easiest of trials to demonstrate success in. But that's where we are. The traditional endpoints used for hemophilia trials, annualized bleeding rate, is still what FDA would like to see as far as accelerated approval. Factor activity would have to be rationalized very strongly with FDA with supporting information in order to get that approved via the accelerated approval pathway. Retinal disease is somewhat more simple in that it is usually the ocular diseases have one genetic defect that can be addressed. They also have some very simple endpoints that have been validated and can potentially be used. Vision loss and visual acuity rate and photoreceptor loss are topics that can be chosen to discuss with FDA as a justification for approval. But like I said, you're going to be in talking with the agency, hopefully not any more than you want to, but you're going to have to negotiate with them on clinical endpoints and the size of your database that is acceptable to them. So it's my philosophy is to interact with FDA as infrequently as possible, but with these therapies, I'm afraid I'm not going to be able to get away with that. So successes to date, obviously if you have an absence of a functional protein, as I said previously, that's the easiest thing to address. How much of your gene is needed to reverse a disease is also important to know. This can be done in the lab before you get into the clinical trials. In many cases, you can assess what the level of expression is in those affected with the disease. And then gene addition can also be done both in vitro or in vivo and ex vivo with cell preparations. So obviously the major challenge is that gene transfer therapy results from the increasing efficiency or the increasing the current efficiency. We have many vectors, none of which is all that efficient in transfecting. So improvements continue to be made on the scientific front, and it may be a breakthrough in that technology that allows the field to really take off. Many of these vectors have immunogenicity issues that need to be addressed and controlled in the clinic. And Dana is going to talk about some of the non-clinical aspects that allow you to prepare for that. And then the manufacturing hurdles will improve with time. And on the horizon, gene editing, there's no clinical data to date with these techniques, but in the near future, I expect to see these move into the clinic, and it may be potential for less off-target toxicities if they are really specific and edit only the genes that they're targeting. So with that, I'm going to turn it over to Dana, and Dana's going to talk about the preclinical considerations in this exciting field.
Thanks, David. As you said, I'm going to briefly go over some of the preclinical considerations for developing gene transfer technologies. The preclinical programs that are put together for gene therapy products are, the objectives are pretty similar to objectives for other drug development programs, to establish biological plausibility, to determine biologically active dose levels, and to inform the clinical trial. There is an FDA guidance that came out in November 2013, a preclinical assessment of investigational cellular and gene therapy products, and they provide a lot of general guidance and considerations. And really, it's the science of your particular gene therapy product that's going to drive the types of studies that you need to do. And I'm going to provide two examples as well of some marketed products. Proof of concept studies are typically conducted in either in vivo or in vitro. In vivo, it's important to use relevant animal models that may necessitate using an animal ortholog of a human therapeutic. And the proof of concept studies really should determine pharmacologically effective dose range and to confirm your route of administration is feasible and to optimize a dose regimen. If you incorporate some additional endpoints can be incorporated into these proof of concept studies, such as biodistribution and persistence studies, looking at how your vector and or cell therapeutic is distributing in the body and how long it's there. And then also some initial evaluations of impacts on the immune system, as David had alluded to, can be incorporated into some of these proof of concept studies that can actually help to guide toxicity testing and may give you a quick read on a potential risk-benefit profile. Okay. Toxicology studies are often conducted to support gene transfer technologies for the clinic. In general, they should mimic the proposed clinical trial design and regimen, they should utilize multiple dose levels, bracketing the human proposed clinical dose range. Because these products can persist in the body for prolonged periods of time, multiple specific time points are usually incorporated into these toxicity studies to evaluate acute and chronic and or delayed onset toxicities and the possible resolution for them. And your proof of concept studies and your biodistribution and fate studies are going to help inform these tox study designs. Species selection is actually quite important for, as it is for all toxicities testing, but for gene therapy testing, really the biological response in the animal model that you're using in your tox studies should be similar to that's expected in humans. You may be using genetically modified animals and or large animals in order to have comparable physiology and anatomy to the humans. Typically only one species is used, and oftentimes the only relevant animal model is a model of disease or injury, in which case you can incorporate some safety endpoints into your proof of concept studies. And I'll show you an example of that. Okay. Okay, and so here's some study parameters that would typically be incorporated into a tox design. In addition to your standard parameters for toxicological evaluation, there are going to likely be some product-specific parameters, and I've listed several of them here. Definitely effects on the immune system, whether that's upregulation or downregulation of the immune system, or cytokine release, as well as development of antibodies to either the vector and or the protein product. And ultimately, the goal not only is to identify target organ toxicities, but to also establish a safe dose level for starting your clinical trial. I'm going to go through two marketed products, just their non-clinical development program. They're a little different from each other. The first one, Luxturna, was recently approved and on the market for treatment of patients with vision loss due to biallelic RPE65 mutation. The product uses a adeno-associated viral vector that expresses the human RPE65 protein. And the important thing about this particular product there are two important aspects. The vector is non-replicating, and it has been modified to minimize insertional mutagenesis. Okay, I outline here just a few of the pharmacology studies that were conducted by Luxturna. They conducted in vitro studies in mutant and normal RPE cells, looking at dose-dependent expression of the mRNA and protein, and they did some in vivo studies in mutant mice and dogs. And here they did incorporate a few safety endpoints into their pharmacology studies. They looked for increases in antibodies, which they really didn't have much increases in antibodies, just transient, and any evidence for potential cytokine response. Biodistribution studies that were conducted for this product were conducted in normal animal models. And of importance, the biodistribution was localized highly to the highest in the ocular fluids, which is expected given the route of administration, subretinal injection. And there was very limited distribution of the vector DNA to non-ocular tissues, and in particular, and no detection in the gonads. Toxicology studies they conducted in normal and mutant canines and normal non-human primates, and they looked at a couple of different dose regimens, including the clinically relevant dose regimen, and they had several endpoints that from 3 weeks all the way to 6 months, so they can evaluate early effects and potentially and later effects. They looked at a variety of several dose levels, including the high dose at 10-fold above the clinical dose, and the safe dose was found to be 5-fold above the clinical dose. They did not conduct developmental and reproductive tox studies or genetic toxicity and carcinogenicity studies, and here are the reasons why: limited systemic distribution, low integration frequency of the AAV vectors, and they don't transduce mature sperm. Also, the drug product is administered to postmitotic tissue. And then my last example here is Kymriah, which is a CAR T therapy. It's a CD19-directed genetically modified autologous T-cell therapy for patients with B-cell precursor ALL that's refractory or in relapse. Their non-clinical program was much more streamlined than Luxturna. They had some in vitro studies looking at the CD19 binding domain specificity, and they did in vivo anti-tumor studies in mouse xenograft tumor models. They incorporated select toxicity parameters into those tumor models. And they also conducted cell distribution and persistence in those mice as well. They did some ex vivo genomic insertion site analysis because insertional mutagenesis is always a concern for gene therapies. These studies, their non-clinical program, identified no safety concerns. However, there are some safety concerns that came out in human clinical trials, and this just shows here the cytokine release syndrome and transient neurological toxicity were pretty frequent adverse events in humans, and those were not informed by the non-clinical program. There's a lot of work, I think, now going on experimentally in laboratories to understand what animal models might be best predictive of the cytokine release syndrome and transient neurological toxicity. And I think that's it for mine. I'm going to turn it over to Scott, who's going to talk about CMC.
Great. Thank you very much, Dana, for that lead-in to my section of today's presentation, where I'll be discussing CMC considerations for gene transfer products, and today with this particular focus on aspects of CAR T technologies. Now, hopefully one of the main takeaways that you get from today's presentation is the need for a well-thought-out CMC development plan, because if you cannot manufacture your product, you cannot run your tox studies, you cannot run your clinical studies, and you will not have the necessary quality information for your regulatory submissions. Now, I'm limiting today's talk to a hypothetical CAR T product, just given the wide variety of gene therapy technologies, as David mentioned earlier today, plasmid DNA viral vectors, bacterial vectors, and some of the more cutting-edge technologies with CRISPR-Cas9, and then obviously the CAR T technologies that we'll be focusing on today. So what is CAR T? Just a little primer to get everyone on the same page. Chimeric antigen receptors are engineered receptors with allowing you to impart a very specific specificity to T cells to target very specific cancer types. They're pretty complicated. They're fusion proteins composed of a antigen, if you look on the left side of the figure there, an antigen recognition domain derived from a monoclonal antibody that is generally then fused with stabilizing transmembrane domains, and then a variety of signaling and co-stimulatory domains. The CAR is then encoded into a vector, then used to transform the patient's T cells for that specific tumor type that you're looking for. But how does this all happen? And it's somewhat of a circular process where you isolate the cells from a patient. The cells are then transported to a manufacturing site where they're genetically modified and then expanded. Then the cells are then further processed into an administratable form and then shipped back to the patient. Seems simple, but this is actually a very complicated process that occurs rather rapidly. Kymria, I think, is about a 22-day process, which in modern GMP world is almost unheard of. And the reason why today we're really encouraging the development of a comprehensive CMC plan is because FDA is going to be placing much greater emphasis on the quality portion of your submissions than they ever have in the past. And we know this because they've said so. In a recent interview, Commissioner Gottlieb indicated that, for most standard drugs, small molecule drugs, traditionally the review process is focused 80% on the clinical aspects and only 20% on quality or CMC related. But for gene therapy products, those numbers are reversed. So you can see that CMC development will definitely be in the critical path for these types of products. And we know this concern is not unwarranted. There was a recent study where they looked at CAR T studies and their ability to actually manufacture the product. And in the studies that they looked at, they found that there was up to a 14% failure in being able to manufacture the patient's doses. And when they looked into this, what they found was that the majority of the failures actually occurred very early in the process of just collecting the patient's T cells. And so problems with collection, selection of the cells, purifying those cells, and then the quality of the T cells themselves. So when you're putting together these protocols, you want to be very mindful of the state of the patients that you're targeting and perhaps any other chemotherapy or other therapy regimens that they might be on at the time you're trying to obtain these cells. So because of the complexity of these products, FDA has recently published some additional guidances. David alluded to these earlier. Obviously, you have the traditional IND requirements in 21 CFR 312. You've got the Phase 1 GMP guidance. And then there's 2 of the 6 new gene therapy or gene transfer guidances. 2 are specifically rated for chemistry manufacturing controls. And I'll be talking specifically about the CMC information for human gene therapy INDs today. So starting at the beginning, one of your most critical starting materials is obviously the T cells that you're going to modify. And in the new guidance, FDA is asking that you provide a fair amount of information as to actually how you're collecting these. So any of the devices, reagents you're using, operating parameters, volumes, any enrichment steps that you might be conducting to maximize the number of cells that are needed for your process, any hold times. Obviously, labeling and tracking is going to be very critical to ensure that the cells you obtain get back to the right patient. And one of the big take-homes from the new guidance that is if you are conducting multi-center trials, you want to make sure that you have standardized collection protocols so that any variability that might be imparted due to different collection methods is minimized. So briefly, in order to modify and expand the T cells, they first need to be activated. So in vivo, in a normal immune system, the T cells would become activated by the presence of an antigen-presenting cell, like a dendritic cell, expressing a particular antigen. But when you're trying to do that artificially, the current state of the art is to use something like a antibody-coated bead. So once you've activated your cells and selected them, they then need to be modified. So you take those cells, you expose them to one of several systems, gene transfer systems, maybe viral or non-viral, encode it into the T cell, and then the T cell will then be expressing that particular receptor. Now, as I indicated, there are several methods that can be used to genetically modify the T cells, and there is no right or wrong method. However, whatever method you use, you'll need to justify in your submission. Probably the most common vectors are the ones that have been used in the two recently approved products, and are the gamma retroviral and the lentiviral vectors. These are used because they're capable of very high rates of gene transfer efficiency and very stable, but they are complex to produce, and so they have quite complex manufacturing processes, and we'll be discussing what some of those requirements are in the next few slides. There are a lot of cell banks and viral banks that almost, as you work on these products, you almost think you might work at Wells Fargo, based on the number of banks that you're going to be dealing with. For a viral vector, you need a master viral bank. Now, I'm not going to go into detail about each of the characterization tests. Some of them are very similar to what might be done for more traditional biologic products. But what I do want to mention is that for a CAR T product, in addition to the viral bank, for example, a lentivirus, you also need to manufacture a number of plasmids. So a lentiviral vector can require up to 4 plasmids, structural proteins, glycoproteins, and some viral accessory proteins. Each one of these plasmids is basically a product in and of itself. It needs to be manufactured, tested, packaged, released before use. And in the new guidance, FDA does recommend that you do provide a certificate of analysis for each of the plasmids that you might be using for your product. You also need a master cell bank for your substrate to grow your virus. Again, very typical characterization studies, but this is quite a bit of information that is now going into one submission. Traditionally, your submission might just have one cell bank, but here we're almost up to 3 now with your viral bank, plasmids, your substrate bank, and you haven't even begun to modify your T cells yet. So as you're modifying your T cells, you're expanding and modifying them, you will need to describe in detail your modification and expansion process. So FDA is going to be looking for things like the specific bioreactors you use. There's a number of different types. Each one has their own intricacies, and you want to make sure that you've indicated that the one that you're using is most appropriate for your process. Batch size and then also what the process is capable of in terms of inputs and outputs, and then obviously control of materials. Your products are going to be touching a lot of materials during the course of their journey from the patient back to that same patient. So you're going to need to include in your submissions list of all the materials used in the process and a description of their quality and suitability for use in your process. If you don't have C of A's, make sure you try your best to obtain them, because certainly justifying their use in your process is going to be critical to FDA. Then, once you've expanded your cells, they need to be washed and concentrated and reduced to an administratable volume. If you're cryopreserving them, you might want to make sure you have a freezing protocol. The product is then going to have to be release tested and then transported back to the patient. In your IND, you're going to have to list your drug substance and your drug product specifications. Obviously, for these products, testing is very rigorous, but and it'll be difficult initially to obtain data as to understand what type of specifications to assign to this product. Hopefully, you're starting to generate data for your non-clinical and perhaps technical batches, so that information can form the basis of your preliminary specifications. And FDA does realize that these tests might be in flux at the time of your IND filing, but you just want to make sure that however you are testing your product, you're providing pretty robust descriptions of those methods in your IND. And here's just an example of the product test for Kymriah. This list I obtained from the summary basis of approval for that product, which is freely available on the FDA's website. And I really encourage anyone who's developing these products to look at those documents, because even though they are heavily redacted, there is still a wealth of information that might help you both on the non-clinical and regulatory side, in addition to the CMC side of your program. In terms of methods, typically not required for phase 1, but you want to make sure that whatever methods you are using are appropriately qualified. They do ask that you have your dose assay qualified before initiating any dose escalation studies. And FDA is asking for quite a bit of information for how you are qualifying these methods, quite a bit more than what would be required for a typical small molecule. But one of the real important take-homes is making sure that you have the ability to compare your preclinical and clinical material in case there are any changes in your process or methods. So you either have the same method, or hopefully you retain some material so that you can make sure that there's no critical changes between that material, so that you're able to bridge your non-clin to clinical. And obviously you'll need stability data. If you're shipping back to a clinical site, make sure you're able to support your shipping conditions. Make sure there is a chain of custody from the manufacturer back to the clinical site to make sure the correct material went to the correct patient. And then also to be able to support any handling at the clinical site once it's thawed, and that there's stability data and in-use stability data to show that your product is suitable for the administration sets that you're proposing to use. Dana, I'll hand it back to you.
Okay, thanks, Scott. So just to wrap up, I've got a couple of slides here about some regulatory considerations that are available for gene therapy products. And most of these gene therapy products are, a lot of them are for rare diseases. And so there's the orphan drug designation that's available that has a lot of benefits associated for that if you're for those therapeutics, if you meet the criteria. And then in 2017, the Regenerative Medicine Advanced Therapy designation came about, and that is specific for cell, gene, and tissue engineering products that lead to durable modification of cells or tissues and are intended to treat or modify serious or life-threatening diseases or conditions. The designation is based on meeting those criteria and providing preliminary clinical evidence. There's a fair amount of benefits available. I think the commitment from the FDA, and including their senior management, to provide some intensive guidance for efficiently developing your program is a fantastic benefit. And then just an example, Kymriah had breakthrough therapy designation that occurred prior to the RMAT designation, and it also offers similar benefits, so that may also apply. And then just one more thing to talk about. This is also from the summary basis of approval. There is oftentimes with these gene therapy products, there's a lack of long-term safety data. The clinical trials are only so long, and so FDA, of course, is interested in ensuring safe use. And in particular for Kymriah, the potential for cytokine release syndrome and neurological toxicity necessitated post-marketing commitment to manage those. And their post-marketing commitment included training and assessment of the sites that dispersed to this product to patients, and also included that those sites had to have at least 2 doses of tocilizumab. It's a monoclonal antibody against the IL-6 receptor, and it's used for treatment of cytokine release syndrome. And that's I think those are just highlighting a couple of the regulatory benefits and considerations.
So with that, I'd like to thank Dana and Scott, and turn it back to Joe for any questions that the audience might have about these different disciplines.
All right, thanks. Before we move on to the Q&A, I'd like to remind the audience that there's still time to submit questions using the Q&A button at the bottom of your screen. We already have a lot of great questions, and we'll try to get to as many as we possibly can with the time remaining. So this first one I'll just kind of throw out. The question is on Chimera. In retrospect, what could have been done that might have helped ID those tox issues ahead of clinical use?
Well, that's a good question, because I think that question has come up repeatedly in the literature, and obviously the animal models that were used, the xenograft tumor models in mice, were not predictive. There's been some studies that have been conducted in primates that look like primates may actually be able to mimic, I think they use CD20 therapies, so the primate models did mimic the cytokine release syndrome and neurotoxicities that were seen in humans. So, but I still think it's a very active area of investigation.
All right, Dan, this next question is for you again. Is it true that CyBER is now recommending large rodent studies over small, large animal studies to get a tox readout?
I'm not aware of that. I think CyBER is very interested in making sure that your animal models that you're using are relevant. They have to have biological relevance. They have to be physiologically relevant. They have to, as best as possible, be able to produce the same effects that you're anticipating in humans. That may require that you use genetically modified animals and or diseased or animal models of injury.
Yeah, and this is David. I just would take any pan-center recommendation with a grain of salt, as there are very few center recommendations that come down on development programs.
Right, yeah, it's scientifically justified, and every animal model that you're going to choose will have some limitations. It's important to understand those and actually provide your justification for your species in your IND.
You should be going to CyBER with your recommendation of a species, not waiting for CyBER to tell you.
Okay, next question from audience member is, what are your thoughts on mRNA as a therapeutic modality and the use of lipid nanoparticles as a delivery system?
Don't have enough experience to be very knowledgeable in that area, but I think it's an up-and-coming area. I think there's been a lot of success with it, and if it's working, I would not discourage you from pursuing using that vehicle.
I think just the similar, evaluation. I think with the mRNA therapy, there's less than potential for insertional mutagenesis, right?
Yeah.
I guess stability and
and persistence of effect with mRNA.
That's right. That's what I'd be concerned about.
Yeah, and I know the liposome studies that I've been involved with. You're not always getting the exquisite targeting that you're now able to get to with some of these newer technologies, specifically CAR-T, where you're able to, it's rather than a magic bullet, it's a laser beam going after a specific tumor type. And at least some of the liposome products that I've worked on before, we weren't really sure where the product was going sometimes, and it didn't always seem to make sense with what we thought was going to happen. So.
Okay, so this audience member says they're currently developing a novel gene therapy program, and the question is, are there opportunities to discuss the non-clinical and clinical development program with FDA prior to a pre-IND meeting?
I'll take this one, or at least and maybe you guys will want to jump in, but yes, there are opportunities. The FDA is not a consultant. So if you can get expertise from either your team and or KOLs or consultants, then please do so before going to the FDA. However, some really novel therapeutic products really do need to be discussed with FDA so that you're building a program together with them. And they do have availability of a pre-IND meeting that you can map out. And it is really there are, for example, CRISPR-Cas9, I think, is a very good example of a novel technology that the Nonclinical Development Program was really, for some of these technologies, are really developed hand in hand and is very specific for each technology. So, yes, there are opportunities.
Yeah, and I just want to emphasize that FDA learns about these technologies from the industry. So you have the experts on your team, and so you can actually approach FDA, which what Dana called a pre-IND meeting, but it's actually you educating FDA about your product and teaching them the science, because they don't have the science that many of the biotech companies do.
Yeah, and just to tag on, some years ago I was involved with a gene therapy product where we did have a pre-IND meeting, and it was very a crucial meeting in helping us map out our non-clinical program, because it was quite a complicated product, but the input we got was invaluable because the program that was designed was acceptable to the agency upon review of the IND. In addition, we were able to get some clarification on a number of CMC aspects related to specifications and also what they wanted to see in terms of the process itself. So highly recommend for these type of products, if you are eligible for a pre-IND meeting, to take that opportunity to interact with the agency.
The next question up is, are there specific animal models that can predict cytokine release syndrome in humans?
I think similar to an earlier question, that's a very intense area of research. There appears that there may be some non-human primate models that have demonstrated that they can potentially mimic the biology and effects of some of these, like a CAR T therapy. But I think it's just a beginning an area of interest. And of course, that doesn't preclude you one from investigating the impact of your therapeutic agent on the immune system in your animal models.
So how important is it to evaluate potential for anti-drug antibodies in non-clinical studies, and are they useful for predicting antibody generation in humans?
Typically, I think they can be very useful for animal models for a couple of reasons. One is because it helps you to interpret the data that you have. If you have a persistence issue or you lose efficacy over time, it's less durable. And you identify these in the animal models, it'll help explain the animal models and the data you're getting from those. How does it translate to people? Not so well. I think that anti-drug antibody data in animals is definitely, you should be doing that in your tox studies for sure. It doesn't necessarily translate to humans, so you'll have to do the same thing in humans and you may get something different.
What is the payment landscape for these therapies, and how are insurers and pharmacies making decisions regarding payment?
Well, that's still being worked out in Europe and the U.S. And actually, I just saw a recent proposal from Medicare that they would reimburse after the patient was cured. So some of these products are so expensive that they're just reluctant to pay for these therapies before they know that there's going to be benefit. And, I think that is a topic that we could probably give an entire webinar on. But needless to say, that the environment is shifting.
All right. Is there variability between the required follow-up duration for subjects in clinical studies receiving different gene therapies, for example, cell-based therapies?
Yes, I think, as we talked about, it depends a lot on the durability of the effect that you've demonstrated. FDA is going to want to see longer for some of the more long-lasting, durable treatments like cell therapy, as opposed to some of the more immunogenic, sensitive vehicles or vectors that are short-lived.
Yeah, I think some of the CAR T products, Camaro, for instance, they detect that over out to a month after administration. So some of these are quite persistent.
And some of them will last like quite a long time for like Kymria, and that's the REMS programs are good at, so that they can actually, if there's therapeutic benefit and they've evaluated it for a reasonable amount of time, get the market on the product if there's a benefit risk there. But then the REMS programs for evaluating, for managing those potential toxicities
and
is important.
I'm sorry to interrupt, Dana, but that's part of the reason why these programs are being treated as continuous administration. So phase 1 morphs into phase 2, morphs into phase 3, or a registration data, and they're going to want to see what the durability is of the gene in the initial subjects that were treated with these vectors, so that they have some sense of how long they are needed to be monitored after the product is on the market.
Yeah, and some of these vectors will further expand in the body. So the dose that you administer is not necessarily, they might actually be receiving more dose because they're expanding in vivo. So
it's very therapeutic specific, but in general, there's long-term safety that's going to be followed, probably mostly post-marketing.
It's data dependent and negotiation dependent, how good a negotiator you are.
And it's all about safety.
Yep.
So next up is, what is the breakdown for institutional clinical sites between academic centers and state-sponsored hospitals?
Yeah, so as we spoke about at the top, these technologies are generally coming out of academia. A lot of times the technology is transferred to a biotech company that's spun out of academia. So, and the physicians at academia are much more used to working with novel therapies, whereas you can maybe find some aggressive community hospitals or state hospitals that are willing to take the risk on of administering these products. But unfortunately, I think we are left to deal with the academic centers primarily and their protracted IRBs.
Yeah, and also to dovetail off that, the study that I had in there showing the manufacturing failures, in that paper, the majority of those studies were conducted at academic sites. And so, you're seeing the variability or the variation in how these products are being manufactured is leading to variation in quality at the end. So with the transfer from academia to more biotech companies, hopefully that variation goes down as companies that are more in tune to what FDA is looking for in terms of manufacturing, that the variability will go down and the success rate will go up for these products.
It looks like we have time for one last question. Do you know if the FDA is going to publish any more disease-specific guidances for gene transfer products?
I would hope so. I think that's the only way that we're going to really move some of these products forward quickly. That has been the case with biosimilars. I think you can look at the approvals of biosimilars in Europe where they do have product-specific guidances, whereas in the U.S. FDA has only published generic guidances. So that specific advice where they coalesce information that they've learned from other sponsors that have submitted INDs, FDA is able to comment much more intelligently in a specific way for difficult therapies such as these.
Yeah, and I think it's such a hot topic at FDA right now. For instance, the CMC for IND gene therapy guidance had been put out earlier this year and has already been the draft guidance has already been updated, very recently. So that seems remarkably fast for FDA. But I think when you just look at the remarkable efficacy that some of these products are providing, it just makes sense for the agency to put this on the front burner and get these guidances out. So hopefully there'll be some more in the future.
All right, that's going to close out our Q&A session. I'd like to thank everyone for attending this Fierce Markets webinar and submitting so many great questions. I'd also like to thank our speakers for participating and Ro for presenting the webinar. Just as a reminder to the audience, the webinar has been recorded and you will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thanks again for joining, and we look forward to seeing you at future events.